Welcome to a highly sensitive, versatile, non-optical assay platform for analyzing low-abundance proteins. I'm Tamlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and I'll be the moderator for this webinar. Quantitative proteomic assays are known to be challenging, but they don't have to be anymore. According to our speakers today, a new acoustic assay technique from Bioscale allows rapid design and configuration of highly sensitive assays to detect and quantify proteins in complex biological samples. If you want to hear more, stay tuned. Our two speakers will explain the Vibe instrument platform and give an example of its use in a minute. First, let me introduce them, and then we can get the webinar underway. Our first speaker is Elizabeth Gardner from Corollas Therapeutics. She will talk about her research on a novel pro-inflammatory protein complex and her efforts to reliably measure the amount of protein heteromer present in human samples. Elizabeth will be followed by Martin Latterich, CSO at Bioscale, who will wrap up the presentation with an in-depth look at the Vibe platform and also provide tips on how to transfer your ELISA, Western blot, or IHC assays to the Vibe acoustic assay format. Before we get started, though, I just want to encourage you to submit questions for our Q&A session at the end of the presentations. We will answer as many questions as we can. Simply type your question into the Ask a Question box and hit Submit. All right, Elizabeth, we're ready for you.
Thank you, Tamlin, and thank you everyone for attending this webinar. Today, I'm here to talk to you about a novel assay that we've developed on the Bioscale Vibe platform to look at a pro-inflammatory protein complex. Corollas Therapeutics has a novel biotherapeutic called CD2009. CD2009's target is a complex between two chemokines, platelet factor 4 and CCL5. These proteins form a complex in the presence of each other, a very high affinity complex. This occurs when platelets are activated and secrete the two proteins. In the case of certain disease states, these two proteins act to cause tissue damage. Our goal is to reliably measure the amount of this protein complex present in human samples and use this specific assay system to rationally guide the therapeutic use of CD2009, which disrupts the protein complex and suppresses the inflammatory damage done by the two proteins complex together. Activated platelets lead to an inflammatory state and actually cause an endless cascade of inflammatory system. Platelet factor 4 and CCL5, when together, promote leukocyte attachment and leukocyte migration, inflammatory cytokine production, and this inflammatory cytokine production actually causes tissue damage. CD2009 has been rationally designed to disrupt the cycle of inflammation and breaks apart the heteromeric complex. How is this specifically related to lung disease? In the case of lung inflammation, platelet activation occurs. RANTES and PF4 are secreted and form the heteromeric complex. This induces neutrophil stimulation and the production of things like elastase. And when elastase and other chemokines and cytokines are produced, this causes leukocyte recruitment. In specific lung diseases such as asthma, COPD, cystic fibrosis, ventilator or mechanical induced lung injury, and alpha-1 antitrypsin deficiency, which is a genetic disorder, you see heteromer-mediated biology come into play. CD2009 is a therapeutic that can potentially help. The CD2009 target, the heteromer, is correlated with elevated neutrophils in the bronchoalveolar lavage fluid of patients with acute lung injury. This particular assay that you see here on this slide was done using a complicated immunoprecipitation technique in the bronchoalveolar lavage fluid, and this was actually published by Grams et al., some collaborators of ours, in 2012. We really wanted to be able to explore the presence of the heteromer and rationally guide the use of CD2009 in a wide variety of different patient samples. We had some idea that the PF4 RANTES heteromer could be measured in vitro. We used Bioscale to do this. We were able to measure concentration-dependent interactions between RANTES and PF4, and we were able to show that CD2009 can disrupt this interaction at low nanomolar concentrations. Intriguingly, the calculated affinity for the RANTES PF4 complex was higher than RANTES or PF4 homodimer interactions. However, using immunoprecipitation or using Biocore is not a very good way to assess the presence of the heteromer in patient samples. We tried a couple of different techniques, ELISA techniques, to actually tease out the heteromer in various lung disease states. We didn't have much success with this, and although the data was encouraging with immunoprecipitation and Biocore, we really needed a more cost-effective, high-throughput way to reliably examine large patient cohorts. To this end, we explored the use of the Bioscale Vibe as an assay platform and engaged in assay development projects with Bioscale. Corollas has collaborations with multiple clinicians to assess patient samples. And the goal of this effort is to really be able to use the assay as a surrogate marker for efficacy of CD2009 use. Ideally, we'd like to be able to put the Bioscale Vibe into the clinical trial and run it alongside of the clinical trial to assess if CD2009 is being effective in patients. It also would guide the use of CD2009 because we don't want to use it unless patients have high heteromer. The Bioscale Vibe acoustic assay technology, which Martin Ladowicz will talk about in a minute, is a very novel platform. It allows you to use your own antibodies, and because we were dealing with a protein complex, we needed to use a specific antibody for PF4 and a specific antibody for CCL5 to be able to measure the complex. We were also working with a wide variety of biological matrices, including bronchial alveolar lavage fluid, sputum from the lungs, plasma, and serum. And so we wanted to be able to address these different biological matrices in an assay platform that was fairly easy to use and didn't require any sort of optical detection that may be disturbed by the different matrices we were using. The acoustic assay technology actually uses a magnet and shear flow and actually measures amplitude. So both PF4 and RANTES are very small proteins. So detecting them on the membrane would be very difficult. The antibodies actually recognize the different proteins in the complex and cause the whole complex to be heavier. So what you actually see, and if you look at this graphic, it shows it quite well, you see an actual change in amplitude by virtue of the fact that the complex is sticking. This is our initial assay, and as you can see, we saw a nice dose-dependent, measurable heteromer dimer that was formed in vitro. And without going into which antibodies that we used, what I can tell you is that we were specifically able to come to a unique antibody pair within the span of 3 days. And as you can see, it's a very nice curve here on the right-hand side. We did some assay development with different antibodies, and our initial antibody pair optimization was very profitable for us. What you see on the left-hand side is recombinant protein curve, and as you can see on the right-hand side, the standard deviation coefficient of variation for this particular antibody pair was quite nice. And in fact, the CVs are consistently less than 10% for a given sample across a plate and across different plates. So this was very encouraging for us that we could actually move into patient samples. We explored plate-to-plate variability. Again, this is with the recombinant heteromer, and if you see, this is plate 1 and plate 2. Very nice linearity across the plates, and the signal-to-noise looks quite nice. Exploring assay variability from day to day was absolutely essential if we were going to move this into the clinical setting eventually. So what we did was we ran the heteromer assay 2 non-consecutive days on 2 different cartridges. And Martin will actually talk about how this is put together and how the use of the cartridges is absolutely fundamental to the use of the Bioscale Vibe, and actually makes it a very easy and low variability way to run the assay. So these are actually patient samples, and as you can see, the day 1 average versus the day 2 average. The same samples were used on both days, and the dilutions were done the day of the assay for each run. All the buffers were made fresh each day, and this is actually hand pipetting. So different cartridge, 2 different days, patient samples, and what you can see is that these are very close. This is actually my hand pipetting, so I'm pretty pleased that the assay looks as good as it does. So the key antibody reagents were from the exact same stock, and this was important to us as well, because what we want to be able to do is go in with the same batch of antibody reagents as we move across a clinical trial or actually assess in the clinical setting whether or not patients are suitable for CD2009 usage. So this is our initial assay run, just looking in a blinded fashion at healthy patients versus sick patients. And with our variety of different collaborators, they all have different techniques and different ways of pulling plasma, getting sputum, etc. So what we wanted to do was actually check across a wide variety of sampling whether or not we were seeing consistent results. So this is a smattering of COPD patients, alpha-1 antitrypsin patients, and also acute coronary syndrome patients that we were working with from 3 different sites, 2 in the U.S. and actually 1 overseas. And this is normal patients from these collaborators as well as unhealthy patients from these collaborators. 18 out of 20 patient samples were above 5,000 nanograms per mil when you actually ran the calculation to look for heteromer. 18 to 20 of the normals were below 3,000 nanograms per mil. We did a couple of different dilutions. We can actually open this window up quite a bit. But this was the initial assay run, and we were completely delighted, except for this one poor healthy guy who's up around 8,000. And we really want to find out who that person is and make sure that they're okay and actually healthy, because now that I've run thousands of samples, I feel very confident that when the heteromer is present, it's actually not very good for the patient, even if they are presumably healthy. So the end goal is obviously to use this assay as a way to check to see if CD2009 is going to be effective in a particular patient population, in a particular patient, really drive the personalized medicine aspect of this. So can the assay actually allow us to see that? So this is an assay that we ran a quick test to see if CD2009 can block the heteromer formation in vitro and reliably be measured in the Bioscale assay. And what you see is that this is very nice blocking. In the blue, you see PF4 plus Rantes, and this is actually in the absence of CD2009. On the red side, you can see this is in the presence of CD2009, the same sample, and this was run multiple times. And as you can see, the CVs here, standard deviation is quite nice. Looking at this assay to see if we can actually correlate high heteromer levels with potential disease state. This is about 300 patients. On the right-hand side, you can see patients that have lung obstruction. So less than 70% FEV over FVC, and this is the forced expiratory volume calculation. This is the Global Initiative for Obstructive Lung Disease standard, the gold standard here. You can see that there's a big difference between healthy patients in the blue and patients that have lung disease in the black, and even between patients that are greater than 70% FEV and patients that are less than 70% FEV over FVC. There is a difference, and this is actually significant if you run the statistics on it. So we didn't anticipate to see that the heteromer would be so predictive of this, and we ran this in a completely blinded fashion with one of our collaborators, and it was quite nice to see that we can actually pick up lung obstruction with this assay. So as a quick project summary, the Corliss heteromer detection assay was really, the whole project was born because we needed a way to consistently detect the PF4 Rantes heteromer in patient samples. We tried a variety of different ways to confirm that the heteromer existed, including via core using standard, surface plasma resonance techniques. We tried immunoprecipitation in actual human samples, and those were both very encouraging. But when we moved that into an ELISA platform, and we actually tried to build a robust assay that we could reproducibly measure heteromer levels in, we were not able to see the heteromer. The Bioscale Vibe came along as an assay platform that we might be able to use. Because of the rapid assay development allowed with the Vibe platform, we were able to produce an assay within 3 days. Very easy antibody binding pair selection, as you can see from the data, very convincing to us that it was going to be a reliable assay straight off the bat. Very flexible regarding biological matrix. What I didn't show you here was the actual data from our sputum collaborators, but that data is very exciting. In fact, what we've seen in raw sputum as well as solubilized sputum is that we can predict exacerbation and actually track exacerbation in this assay. which is very exciting to us, and that the heteromer actually tracks in this assay with other markers. So there are sputum biomarkers that have been published on. The heteromer is not one of them at the current time, but if you correlate markers of inflammation with the heteromer in this assay, you actually can get a very nice predictable correlation. The acoustic system is very nice. It uses an 8-channel system, as Martin will tell you later, rather than using a 96-well platform or a 384-well platform. This actually lowers assay variability, and the fluidic nature of the assay actually allows you to have both the flexibility in the matrix as well as flexibility in your overall pipetting. Corliss is now actively measuring levels of heteromer in patient samples with all of our collaborators, and we're looking at lavage fluid, sputum, and plasma, and serum. We're actually working to validate this assay across a broad range of different disease states. So thank you very much for attending, and please, if you have any questions or you would like more information about the assay or our therapeutic, C2009, please feel free to email me. This is my email here. And now I'm going to actually hand it back over to Tamlin to introduce Martin.
Thanks, Elizabeth, for a great presentation. You're a tough act to follow, but I'm sure Martin is up to it. But Martin, before you get started, I just want to remind everyone one more time about the Q&A session at the end of the presentations. All you need to do to submit a question is type a question into the Ask a Question box, and we'll answer as many of those as we can at the end of the presentations. All right, Martin, we're ready for you.
Thank you, Tamlin. Good morning, and thank you all for tuning in. I'm Martin Latteridge. I'm the Chief Scientific Officer at Bioscale. And after Elizabeth's outstanding presentation, it's my real pleasure to introduce you in some more depth to the Bioscale acoustic assay technology that enabled Elizabeth to develop a heteromer assay and it allowed her to carry out the retrospective patient study. In my presentation, I will cover the acoustic technology and how it's enabling fast assay development at our users' labs. In addition, I will show data from some of our current customers, such as some typical cytokine assay characteristics in serum and plasma, some comparative case studies that demonstrate superior analytical performance of our acoustic assays over Western and ELISAs. Also how our clients use our signal transduction pathway assays to characterize cancer cell lines in search for treatments of KRAS-dependent cancers. And lastly, a case study from Takeda Pharmaceuticals that demonstrates the utility of acoustic assays in samples that are refractory to analysis by conventional ELISA assays. So first, I would like to go into some more depth into the actual assay format. And so what you see on the left panel is when we actually perform an acoustic assay, we do a pre-assembly of a capture antibody, analyte, and detection antibody complex. So the way the assay is carried out is we take magnetic beads that have a coupled capture antibody, which allows it to capture analyte from the mixture. We also have a detection antibody that has a small hapten conjugated to it, which allows the formation of an antibody sandwich in case you have analyte present in your sample. And so this entire complex is pre-assembled in a pre-incubation time when you incubate your biological sample with our reagent mixtures. We then automatically transfer the assay mixture into a cartridge which contains the acoustic sensor. And this sensor has a anti-hapten which allows it to interact with the hapten and lead to complete formation of the bead to surface. So when we introduce the assay mixture into the cartridge, we engage a magnet which pulls all the magnetic beads to the surface of the sensor. Then we engage a liquid flow which takes away all non-specifically associated beads once the magnet has disengaged, and you're ultimately left with only specifically associated beads. So then, when you have an acoustic sensor that vibrates at a high frequency of 25 megahertz, and you attach weight to the surface, you end up with slowing of the vibration, and this slowing of frequency is actually directly related to the amount of mass that was originally deposited on the sensor surface. So if you look at the bottom right panel, you actually see a trace, an acoustic trace, what actually goes on the sensor surface. So on the left, you have no beads associated. When you engage the magnet, all your possible beads associate with the surface, which you see as a huge spike. And then once you disengage the magnet, all non-specifically associated beads dissociate, and you're actually left with only specifically associated beads, and that can be translated directly into a protein concentration signal against a standard curve. Now, the benefits of doing acoustic assays are, firstly, because you're dealing with an acoustic and non-optical system, you can actually analyze your favorite biomarker in complex matrices, and you get very competitive performance, as I'll show a little bit later, that actually is superior to your ELISAs and Western blots that you might be doing in your labs. So it allows you to directly measure in complex matrices, even the types of matrices that are known to cause optical interferences, such as plasma and heme-containing samples. The actual assay development, as Elizabeth mentioned, is very rapid. So very quickly are you able, by using your own antibodies, you're very quickly able to develop a novel assays. And what Elizabeth just now mentioned was actually an assay that was only developed in 3 days of experimentation. Just in comparison, a similar assay developed by traditional means on an ELISA would take significantly longer. And so the system is, of course, automated, and so it allows you to very quickly screen through conditions or alternatively analyze significant numbers of your favorite samples. And so just as a reference point, in Elizabeth's study, she's been looking at thousands of individuals by now, which it just demonstrates the kind of throughput that you could accomplish on our Vibe system. And assays have exceptional dilutional linearity and significantly reduced running costs, predominantly also in part because you're actually using less antibodies in the system. So it's actually great for saving your precious antibodies. And so in summary, the Vibe system is an enabling tool for every protein lab. It enables new assay development for users in biomarker development labs. And you would recover actually the instrument cost in less than a year. And the comprehensive cost of an assay is below $4. So it's actually very competitive with when you compare it to other types of technologies. In terms of assay configurations, our users generally carry out two principal assay formats. So on the left panel here, you have a sandwich assay format. And so the sandwich assay format uses two cognate antibodies. So you have your capture antibody, which is attached to the magnetic bead. You have your analyte, and then you have a cognate hapten-tagged detection antibody, which allows the interaction with the anti-hapten surface should a successful sandwich have formed in the presence of analyte. So the benefits of the sandwich assay is it is very sensitive, it's very specific, but also due to the format, it allows you to do different things. For example, it allows you to measure post-translational modification of your favorite analyte, such as phosphorylation. On the right-hand side, you see the competitive assay format, which is typically used when you only have one antibody available, or when your analyte has a single epitope, or when your analyte is a small molecule. And so the benefit of carrying out a competitive assay on the Bioscale platform is that the assay is several orders of magnitude more sensitive than a comparable competitive assay by ELISA, but also allows you a great flexibility to actually measure different types of analyte in your sample, provided you have specific reagents, specific affinity reagents available. And to show an example of the beginning of an assay development project, so the first thing you would do is you would actually test different combinations of antibodies to see which antibodies show superior pairing in the development of an immune assay here. And so what is shown here is an example of a P38 assay that was developed at Bioscale. So in brief, what you do is you take different combinations and orientations of antibodies in the presence of analyte, and you interrogate to see which antibody combination gives you the best signal-to-noise ratio. Now, this type of assay development step is significantly simpler than what you may be used to when developing your own ELISA assay, because actually the checkerboarding on our acoustic assay platform has many benefits over traditional ELISA assay developments. For example, we require 10 to 100 times less antibody per well, and we don't have to do the extensive titrations that you would have to do when doing ELISA development. Also, we can directly test for antibody pairing in relevant sample matrix, and something that would be very, very difficult to do in ELISA assays or even other techniques such as surface plasmon resonance-based techniques. So just as a reference point, Elizabeth's heteromer assay, which was one of the more sophisticated assays that was developed, actually could be accomplished in 3 days. So in the next section, I wanted to go into some concrete examples of different types of assays that have benefited our users. And I'd like to start actually with a detection of biomarkers in serum and plasma. And a good example here is shown for a human interleukin-6 assay in serum and plasma. And so that's an assay that was developed specifically for measuring the cytokine in some of the more challenging sample matrices, such as hemolyzed plasma and amniotic fluid. And so on the top left panel, you see a typical composite calibration curve. On the top right panel, you actually see some of the assay characteristics, such as the lower limit of detection for this specific assay is actually 140 femtograms per ml, both in serum and plasma. The average spike recovery, which is a measure of assay interference, shows values both in serum and plasma that are very close to the ideal 100%. So in this particular case, if the normal values for good assays is considered somewhere between 80 and 120%, and so you see that this assay compares extremely favorably to these assay principles. And so the assay is actually sensitive enough to measure the normal levels of interleukin-6 in 12 out of 12 serum samples. And as Elizabeth mentioned earlier on, the actual CVs for this particular assay are very, very favorable. So in the picogram per ml sensitivity range for this assay, they are at or below 5%. And in the femtogram level, they are actually also very competitive. So the benefit that our users have seen when they use an acoustic platform is that they get a great day-to-day reproducibility as well, which allows them to consider our platform as a platform of choice for longitudinal type studies. Shown here are some downward dilutional linearity curves of a acoustic assay compared to a ELISA assay, and these data are data for a different cytokine. And generally, what you expect is if you have an immunoassay and this assay shows little to no interference by other plasma or serum constituents, you expect if you have different individual patient samples, you expect them to dilute in a linear fashion, and also the comparison of the linear dilution curves should be close to an ideal parallel to each other. Now, when you compare actually the acoustic assay curve versus a ELISA for the same analyte, what you notice is that for this example, the acoustic assay was significantly better, showing close to ideal dilutional linearity when compared to the ELISA assay that actually showed at least two patients that had interference, that were actually interfering with assay performance. So this is actually very important to investigators when they need a accurate measurement of the analyte they're measuring, because ultimately you want your analytical signal being due to the presence of analyte and not due to the presence of a assay interferent. When you compare acoustic assay performance to an ELISA, the sort of general observation is, as is shown here from some data from a Stanford investigator, that showed a 10 times greater sensitivity of the Bioscale acoustic assay compared to a equivalent ELISA assay. And so for the markers AppCam and CD166, you could actually see a boost in sensitivity. For AppCam, it's actually 20 times, and for CD166, it is 10 times. So overall, your sensitivity is greatly improved, and so that becomes important when you're looking at some of the analytes that may be present at very low concentration. When you compare the actual assay performance in cell tissue and tumor lysates, you also see significant benefits. For example, when you go into the Western blot and compare a Western blot against a receptor tyrosine kinase in cellular extracts, what you see is it took actually a lot of sample, meaning 10 micrograms of cell extracts, and very long exposure conditions of the ECL Western blot. And you actually see two bands here in the bottom panel that are very, very difficult to accurately quantify. In comparison, on the top panel, you see the result of a Bioscale assay that measures the same receptor tyrosine kinase, but here the benefit is you actually get away with using a quarter of the material that would have been necessary for the Western blot, but you actually get a very nice quantifiable data. And this type of data reflects basically the presence of recombinant control and then the presence of the actual receptor tyrosine kinase in the cellular extracts. And so you actually see from the top panel, it's actually very easy to accurately quantify the concentration of the receptor tyrosine kinase in these samples. But more importantly, you also see really great CVs as evidenced by the error bars. And so the benefit of using an acoustic assay over a Western blot is you require significantly less starting material, and it also gives you a unique ability to quantify minute differences in protein expression. And so an example actually of how could an assay has been used is shown on the next slide, because in addition to the performance features, because our assay is configured in a microtitle plate, it actually gives you great flexibility to develop your own your specific assay layout. And so here, in this particular example, you see a microtitle plate that was configured so it could measure the phosphorylation status of different proteins that are involved in signal transductions. For example, phospho-AKT, phospho-ERK, phospho-P38, phospho-MEC, etc. And so what you can do in a plate layout like here is you could measure all these different phosphoproteins across different sample types, different induction conditions, or different drug inhibitors. So that's a good example, actually, of how users would actually use our available assay kits for measuring signal transduction pathways. And a good example, actually, of a end user that is using this type of assay layout for their own research is actually shown on the next slide. And shown here is an example of an investigator, Frank McCormick at UCSF. He used our signal transduction kinase assays to interrogate the signal transduction status of different KRAS-dependent tumor mutant cell lines. And what this complex set of data shows is it shows the measurement of different phosphoproteins in the KRAS-dependent signal transduction pathways, and it also shows different inhibitors such as small drugs or small interfering RNA inhibitors that were used to investigate ultimately which of these many important signal transduction nodes do you have to ablate in order to see significant shifts in signal transduction activity that ultimately will target the cells into an apoptotic pathways. And so this particular example is an example that shows a relatively simple, straightforward, and quantitative approach to uncover subtle differences in the signal transduction pathway activity, and something that would have been nearly impossible to do by Western blot, because the kind of data you see here is the result of a relatively rapid and straightforward approach, whereas if you'd done it by Western blotting, it would take you anywhere between 600 and 800 semi-quantitative Western blot to derive at the same quantitative type of data set, but then using significantly more reagent and very significantly more time. And lastly, I want to focus on a feature of the Bioscale platform that actually allows us to interrogate or allows us to measure biomarkers in samples that are normally very, very difficult to be used as starting material for bioanalytical measurements. And so if you remember from Elizabeth's presentation, Elizabeth actually outlined some of the benefits in some very unusual sample types, such as sputum, which are traditionally very difficult to measure. But what I wanted to talk about here is actually another sample type that is commonly used in cancer research, but traditionally it's been very difficult to tackle using some of the existing bioanalytical procedures, such as different types of ELISA formats. And so the example here that I want to mention comes from Takeda Pharmaceuticals. And what Takeda was interested in is measuring a pharmacodynamic markers for proteasomal inhibition. And so Takeda had developed an assay to measure GAT34, which is a stress-induced protein implicated in the control of protein synthesis and apoptosis. And ultimately, GAT34 is turned over and eliminated via the proteasome. So if you have the presence of a proteasome inhibitor, you expect an accumulation of GAT34. And so the original assay that Takeda had developed was actually developed in cell extract. And so they had a ELISA-style assay that was actually able to quantify the biomarker in cellular extracts. However, when the company migrated over to a xenograft tumor model, that assay was no longer able to perform with the same fidelity, because actually the xenograft tumors contained heme that were derived from blood vessels of the tumor. And so ultimately the assay was no longer useful when the model system was changed. And so Bioscale offered the development of a acoustic assay that measured GAT34. And when Takeda compared the performance of the acoustic assay to the performance of their own internal alpha screen assay, the first thing they noticed was the acoustic assay was able to boost the level of sensitivity. So you got about 10 times more sensitivity by using the acoustic assay. But equally, if not more important, by using the acoustic assay, which is shown here on the right panel, you could actually see that the acoustic assay allows the measurement of GAT34 in xenograft tumor extracts, whereas on the left panel, with their own traditional method, the alpha screen was actually not able to measure in tumor extracts because the type of assay is inhibited by the presence of heme and iron. So that's an example of why you would want to use an acoustic assay over an optical assay, because there are instances, for example, hemolyzed plasma, tumor extract, tissue extract, or other types of samples where you may have optical interference that interfere with the actual assay performance. So in summary, what I hope to convey is that the Bioscale acoustic VI platform gives you significant advantages over other techniques such as optical techniques. It's a platform that allows the sensitive quantification of biomarkers. You will not have the same optical and chemical interferences that you commonly see in certain sample types. And also, you can get an assay that is more sensitive and more robust when you compare it to your ELISAs and Western blot. The benefit is it allows a really rapid assay development because you don't have to do as extensive a condition scouting that you would have to do compared to ELISA development. So significantly cuts the cost of assay development, gives you very fast pair selection. It uses diluent, very ready to use diluents that are for assay development. It gives you walk-away type of operation, so it eliminates labor-intensive steps that are common in Western blots and ELISA, so you don't have to worry about extensive wash steps and having to line up in front of ELISA wash stations, etc. the assays give you a dilutional linearity, so that's connected to the robustness of the assay and a single-digit precision. And the running cost is significantly lower than when you compare it to ELISA or Western blot. So lastly, it's an enabling tool for every protein lab. So and it also allows you some flexibility in migration. So, for example, you can develop an assay on the instrument, and you can also use the instrument to actually carry out the actual analysis. Then, lastly, our contact information is shown below. So anybody who's interested can contact us for an on-site demonstration of the acoustic assays if you send us an email to info@bioscale.com.
All right, so ideally, Martin just answered all of your questions, but if he didn't, you should submit them now. We have plenty of time for our Q&A session, and Martin and Elizabeth are standing by and ready to answer your questions. Before we start the Q&A session, though, I just want to let you know that a post-webinar survey will deploy shortly in the presentation manager. You might need to disable your pop-up blockers to view it. Please do so and take a few minutes to respond. Your comments are extremely valuable to us and to Bioscale as well. All right, we're ready to start our Q&A discussion. We have a lot of good questions already, and we'll answer as many as we can. Our first question is for Elizabeth. Did you test operator to operator to see how robust the system was for using in a clinical setting where different technicians might run the system on different days?
Yes, actually, I tested this in probably the best way possible. I had a college student come in who had absolutely no biology experience at all. So essentially it was teaching him how to pipette. And he was a quick study, but essentially I took one day to teach him the Bioscale assay, and he was able to run it on his own. His standard curve looked great. He ran actual samples the next day, and I think if you can take someone who's completely naive to assay development as well as assay running and have them, running actual clinical samples by the second day, it's pretty phenomenal. So I would say yes, you can. It's pretty robust. So I would hope that at clinical trial centers where you would have people who are very accomplished at this, it would be an easy training.
All right, I have another question for you. What makes this different or better than ELISA?
So from my perspective, we've worked with a lot of different matrices, including the sputum, which is a rather messy matrix to work with. It's really one of the more unpleasant matrices I've worked with. We did very minimal cleanup and prep of the sputum, and were able to utilize it in the Bioscale assay. I know that from other people who have used this assay that heme has not been a problem for them, and so going straight from blood has been a very facile transfer. That's one thing that's very difficult to do with a colorimetric assay. Having heme components, they really interfere. So I think that it's probably better from the matrix perspective. It's also probably, it's just very rapid and easy. And because you're using your own antibodies and you don't have to rely on even commercially available antibodies per se or assay kits that are out there, you have a lot more flexibility. For our particular question, we needed to really hone in on specific parts of those proteins and have an antibody that recognized specific partners. The pieces, truly epitope mapping almost that we had to do with our antibody selection in order to actually be able to capture this heteromeric complex. That's a really good question. Thanks.
All right, and thanks, Elizabeth. Martin, now I have a bunch of questions for you. The first one, is it possible to multiplex?
Absolutely. So the nature of our assay is they are parallel uniplexed assays, meaning each assay is an individual assay. The way you would set it up is you'd set up a microtiter plate, and each well in a microtiter plate would contain an individual assay against an analyte of interest. So what that means is you can multiplex by taking multiple samples and looking for different analytes. You cannot multiplex in the sense of, for example, multiplexed technologies that use the same microtiter plate. Now, the benefit of our parallel uniplexing comes because a lot of antibodies, when they're in a multiplex mixture, they show certain interferences that can distort the analytical performance of the assays. And so the benefit using the parallel uniplexing approach comes, you do not see these type of antibody interferences that you can see with multiplexed assay solutions. So yes, multiplexing is possible, but the multiplexing is carried out in parallel and requires individual samples per analyte.
All right, Martin, I have another question for you. Can you perform development and measurements yourselves, or do you have to have companies, or do you have companies you can point to who could do this if we prefer to outsource rather than bring the technology in-house?
Absolutely. You can, and in fact most customers develop their own assays and then carry out their own analytical assays in their own labs. But we do have international service providers that provide both assay development and assay services. And you can contact us offline for additional information how to get in touch with them.
All right, Martin says, can you please talk briefly about how the hapten binding sensor is refreshed between sample cycles?
Absolutely. So as you remember from the presentation, you have an anti-hapten surface that recognizes the hapten on the detection antibody. So then once we perform a measurement, we add a regeneration buffer that strips the interaction between the hapten and the anti-hapten surface. So you follow an analytical cycle on the sensor surface with a regeneration and a buffer equilibration step. And so effectively that means you can regenerate the sensor surface many, many times. In fact, you can regenerate it so much that you can use up to 6 96-well plates for an individual cartridge. So you can recycle it so many times that you can read out 6 96-well plates.
All right, Martin, here's an easy question. Can a novice to protein assay development use your platform?
Absolutely. Most of our customers actually have people in their labs, like Elizabeth just mentioned, that have very limited assay development experience. And so we provide SOPs and protocols and guidelines that enables an end user to do their own assay development. Just as an example, we have demoed or placed instruments, and we got people up and running within the first 2 or 3 days, and they're able to develop their own assays, meaning that they're able to select the antibody pairs, and they're coming up with actual assays that they can use for bioanalytical purposes. So the ease of use of the system is perfectly suited for assay development.
Okay, Martin, what exactly is the basis for tenfold higher sensitivity of AMMP as compared to conventional ELISA?
That's a very good and interesting question. There are actually a number of reasons that underlie the finding that when you take a typical ELISA assay and you convert this ELISA assay into an acoustic assay, very often you get 10-fold or 20-fold improvement in lower limit of detection. And so underlying that is anything from a sample type. So in certain samples, you have interference that interfere with measurements. So for example, hematine, bilirubin, in certain samples, they affect ELISAs. And because the acoustic assay is fundamentally different, you don't have those interference interfere in that assay. Then you have assay format considerations that come into play. For example, many ELISAs, you use extensive wash processes to reduce the background of the assay. And in our case, we use in principle, homogeneous assay format, and we use a very limited wash process that takes place on the sensitive surface. And so, for example, if your affinities of your antibody is limiting, you would expect to see significantly less analytical performance in an ELISA that sometimes has 7, 10, or even 20 individual wash steps, whereas in our case, you would not see that performance deterioration.
All right, Martin, I'm going to give you a short break, and we're going to throw a question to Elizabeth. Is there much sample prep cleanup prior to sample introduction to the cartridge?
Depending on what kind of sample you're using, no. In fact, for plasma or serum, I've done a dilution step and actually loaded it directly into the assay plate. I haven't had any difficulty with matrix interference, if that's what you mean. As far as dealing with sputum, even raw sputum, it's been pretty minimal. And I'm looking at the person who asked the question, Charlie. Charlie, I'd be happy to send you a protocol. I do have a protocol for sputum cleanup if you're interested in that. And it's very simple. It requires a centrifugation. And it's not too difficult. And we've done it a couple different ways where we've weighed sputum out, and we've actually done solubilized sputum, et cetera. So have a couple different ways of dealing with that. I've talked to some people who've used the Bioscale, and they've been able to have very minimal prep as far as working with whole blood, et cetera, or tissue lysates. Again, pretty simple sample prep. So different than ELISA in that perspective. One thing I forgot to mention is that the dilution buffer that Bioscale provides, we've had excellent success with that. They have a couple different forms of it, and what we're using now for our sputum as well as plasma and serum is amazing. I mean, it's a great blocking buffer, and so it's a nice way to get your sample in good shape and ready for the assay. So thanks for the question.
All right, Martin, we're back to you. What reagents are used as supplied? The primary antibody and hapten-labeled secondary? Question mark.
That depends. So we have a open platform approach. That means we provide the cartridges and the magnetic beads and some information how to label the antibodies. But alternatively, we also have a catalog of already available assay kits for somebody who wants to use those kits for specific biomarker analyses. But ultimately, the customer provides the antibody, meaning the capture antibody, the detection antibody, and the analyte in some instances for as a standard for constructing standard curves. So that is for your own assay development purposes.
Okay, Martin, how many microliters do you need to use for the assay?
In terms of sample, the sample volume is anywhere, and that's for a typical serum or plasma sample. You would use maybe 25 microliters of actual neat serum and plasma per assay, and then you typically would use it in a 1:4 or 1:8 dilution. Now, in terms of tissue extracts, membrane extracts, cell extracts, we can load anywhere from 0.1 to maybe up to 10 micrograms per well. So if you compare the loading or the sample loading compared to a lane on a Western blot, you get away with a significantly less starting material.
All right, Martin, one more question for you, and then we'll go back to Elizabeth. But is there a standard to run together as samples? It depends on the type of measurement that somebody is doing. For example, if you're interested in a ratiometric measurement, so you're comparing two samples and two conditions, as for example is the case in comparative phosphokinase analysis, you actually do not need a standard per se. However, if you want to determine the concentration of a protein analyte in your sample, you would normally construct a standard curve using a number of different dilutions of your analytical standard. So you would
if you want to quantify and want to do absolute quantification, you would compare that to a analytical standard curve.
All right, Elizabeth, this question is for you. Do you run also a standard curve together with samples?
Yes, on every plate. So I normally run a standard curve in duplicate, typical 8-point standard curve. And to kind of get at one of Martin's earlier questions, when we do our heteromer assay, we have to do a pretty serious dilution, 1 to 200, 1 to 300 of plasma or serum in order to see heteromer and actually have it be linear. Otherwise, it's going to saturate and be out of range. With one of the assays that we developed actually on the Bioscale in tandem with our heteromer assay, we needed assays to actually look at PF4 or RANTES alone so that we could make sure that what we were looking at was truly the complex rather than the PF4 or RANTES by itself. And our PF4 assay actually ended up being a femtomolar assay, and so the dilution for that is even steeper. So when we get samples, these clinical samples from our collaborators, sometimes they can only send us 20 microliters of sample. But when you're working with 1 to 200, 1 to 300, 1 to 500 even as your dilution factor, then 20 microliters goes a long way. So that's been a really easy way to get collaborators to buy in to work with us on developing this assay because it's such a sensitive way of looking at it. Much more sensitive than the ELISA measurements that we were looking at when we first engaged in trying to find an assay.
All right, Martin, we're back to you, and this person acknowledges that they missed part of the presentation. So my question is, what would be LLOQ of the assay for cell culture samples for tighter measurements, protein concentration? I guess what would the measurement be? It would be in the micrograms, nanograms, picograms?
So for general measurements using the magnetic bead assay format, you expect the concentration to be in the picogram per ml range for these type of measurements.
All right, Elizabeth, we're back to you. You said the test is not sensitive to sample matrix. Much discussion mentioned pretreatment, dilution, etc. Please explain.
Okay, this is a good question. Sorry if it was confusing. So essentially, the assay is so sensitive, we actually have to make a dilution. And we dilute into the Bioscale buffer, which is actually a nice blocking buffer as well and prevents non-specific interaction. So that's what I mean by dilution. We have to dilute, otherwise we would completely saturate the AMMP membrane, and we wouldn't be able to get anything quantitative out of that. As far as pretreatment goes, for sputum, there are several ways of dealing with sputum, which is a really sticky, globby matrix, and it's really difficult. It's not very soluble. So you can solubilize sputum in a variety of different ways, and I'd be happy to send you some protocols on that if you email me directly. And rather than go through a lot of details, we have a very simple protocol for just spinning the sputum biomass down and actually dealing with it that way. So I'd be happy to send you protocols. Good question.
All right, Martin, we're back to you. Could you please comment on the shelf life of the cartridge if it's not opened from the package, and once opened and after a 96-plate run, time limit within which it should be reused?
So that depends a little bit. Generally, the actual shelf life of the cartridge post-manufacturing is about a half year. Then once you have opened the cartridge, it depends on the type of cartridge packaging that or the type of cartridge that you buy. The shelf life is limited in agreement with how many times you actually can use the cartridge. So generally, we use the 3 plate cartridges over about, well, it's enough for 3 runs that happen usually over 2 days or so. However, each product will have that information available as part of the product insert.
Thanks, Martin. Unfortunately, our time is up. Thanks for all the great questions, and I apologize that we couldn't get to all of them. Elizabeth and Martin have said they will try to answer as many as they can offline, and you should take them up on that. This webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and family. Once again, I want to thank our panelists for participating today, as well as our sponsor, Bioscale.
Thank you, Tamlin.
Thank you, Tamlin. And thanks, Martin.
Thank you, Elizabeth.